This is Stop Motion Cellular Respiration by Abby, Amanda, Jeremiah, and Noah. Cellular respiration is the transference of stored energy within cells into molecules of adenosine triphosphate, or ATP. Cell respiration can occur either with or without the presence of oxygen, termed as aerobic or anaerobic, respectively. Either way, cellular respiration begins with the anaerobic process of glycolysis, which produces 2 3-carbon molecules of pyruvate for every molecule of glucose it breaks down. Glycolysis occurs in the cytoplasm of the cell and produces 4 molecules of ATP, but because its energy of activation is 2 molecules of ATP, glycolysis results in a net gain of 2 ATP molecules. These ATP molecules are produced by what is called substrate-level phosphorylation, which is the direct enzymatic transfer of a phosphate to adenosine diphosphate, or ADP. The amount of ATP produced from substrate-level phosphorylation in glycolysis, however, is relatively small. Much more ATP is produced in the later step of cellular respiration. Before entering the Krebs cycle, the pyruvate generated in glycolysis undergoes the link reaction. This is a process in the mitochondrial matrix, which combines the pyruvate with the vitamin coenzyme A, forming acetyl-CoA, and, as a byproduct, 1 molecule of nicotinamide adenine dinucleotide, or NADH, per pyruvate. It is this acetyl-CoA which enters the Krebs cycle. The Krebs cycle, also known as a citric acid cycle, is a cyclical series of enzyme-catalyzed reactions. Each turn of this cycle, moving from higher to lower potential, releases 3 molecules of the coenzyme NADH, 1 molecule of the coenzyme flavin adenine dinucleotide, or FADH, 1 molecule of ATP, and the waste product carbon dioxide. These coenzymes are required for cell respiration, carrying protons or electrons from glycolysis and the citric acid cycle to the electron transport chain. Their respective enzymes, NAD dehydrogenase and FAD dehydrogenase, both facilitate the transfer of hydrogen atoms from a substrate to said coenzyme NAD+. Without this NAD+ functioning as a proton and electron acceptor, neither glycolysis nor the citric acid function could continue, killing off the involved cell. ATP in this step is also produced by substrate-level phosphorylation. Again, the amount of ATP produced from substrate-level phosphorylation is relatively small. The next final step in aerobic cellular respiration, known generally as oxidative phosphorylation, is comprised of 2 sub-processes, the electron transport chain and chemiosmosis. Oxidative phosphorylation refers to the oxidation of the NADH and FADH2 carrier molecules, powered by the redox reactions of the electron transport chain. Said electron transport chain is a proton pump that combines an exergonic and an endergonic reaction in order to pump protons against the gradient between the matrix and the outer mitochondrial compartment, establishing the proton gradient within the mitochondrion. Consisting of a collection of molecules embedded in the cristae membrane or inner membrane of the mitochondrium, the electron transport chain carries electrons delivered by NADH and FADH2 from glycolysis and the Krebs cycle to oxygen via a series of redox reactions. This highly electronegative oxygen serves to pull the electrons through the electron transport chain. The resulting electrons are then paired off with 2 protons and half an oxygen molecule, forming the final waste product, water. But the protons pumped by the electron transport chain cannot diffuse through the cristae membrane. They can only flow down the aforementioned proton gradient into the matrix through what are called ATP synthase channels. Known as chemiosmosis, this process generates the energy to phosphorylate ADP into ATP as the protons flow through these ATP synthase channels. It is in this process of chemiosmosis that 90% of all ATP is produced in cell respiration. Summing up all of the ATP produced in these processes, around 36 to 38 molecules of ATP can be expected to be released from the aerobic respiration of 1 molecule of glucose. Now, this is where cell respiration will really take your breath away. Without the presence of oxygen, respiration, appropriately termed as anaerobic, involves a catabolic process within the cytoplasm of the cell that combines glycolysis and alcohol or lactic acid fermentation. Given that there is an adequate supply of NAD+ to act as electron acceptors during glycolysis, fermentation can generate ATP. There are 2 types of fermentation: alcohol fermentation or lactic acid fermentation. Alcohol fermentation refers to the process in which pyruvate from glycolysis is converted into ethyl alcohol and carbon dioxide in the absence of oxygen, while also oxidizing NADH back to NAD+. Lactic acid fermentation, on the other hand, involves the reduction of the pyruvate from glycolysis to lactic acid.